Thank you, Professor Jonas, for this excellent talk. So let's start our Q&A session.
Thank you, Professor Jonas. I definitely enjoy your talk, and the title is fantastic, Basics to Advance. I think sometimes our thinking is a bit muddled. My question, I'm not a natural scientist, although I study microbiology as my first degree, but today I'm a political scientist. And my question would be, how much does the environment, air pollution, water pollution, genetically modified food, interfere with the functioning of the ribosomes, especially with their capacity to be accurate and fast?
Okay, I'm not an expert in what you ask, but genetically modified food or whatever does not do anything to the ribosome. Ribosome keeps working. And if I can say something about genetically modified, that the politicians, and the crowd are afraid of it, I think that the crowd and the politicians have to rethink of what's going on. We are all what we are eating before we knew genetics, for the last hundreds of years.
Other question?
All what we are eating for the last hundreds of years is genetically modified. If you think about, you eat only nice red tomatoes, and peas were actually the first plant that was divided to several types by a person. This is all using genetically modified without understanding the genetics. So the fact that humans were choosing this type and enriching this type and not that type does not make it less genetically modified, just make the human not understanding. Now we understand, or we can understand what we are doing, and we can do great things. Why to think that this is bad? Of course, in every society, in every type of work, there are criminals. We cannot avoid this. But because one person is killing another one, all that society has to go to hell? No, it's the same. So if somebody is using genetically modified products for the wrong results or by the wrong reasons, we can talk to him or somehow convince him, but I don't think that there are many of these. I think that if genetically modified crops are made with thinking, and I hope that scientists think, then I don't see why it should be so opposed by the crowd that understanding it nothing. What do you think, that genetically modified food will make all of us mice or chicken, or all the chicken will become elephants? Oh, it's adding a bit of protein to food dull by protein. It's okay. Now about environment, again, the environment does not disturb the ribosome. The ribosome works. It's a machine. It's a factory. You give the gene, it translates it. It may affect the gene. It may cause mutations. And this should be closely monitored. But the ribosome, poor guy, doesn't understand. It does, what it gets, it makes. Okay?
Right. Thank you.
Sorry that if I
very clear.
hurted you.
No,
I said politicians and the crowd.
No, I'm not a politician.
I know that they don't like to hear
political science.
What is the most important quality one should have to become a successful scientist? And yet it can be trained. Thank you.
I don't know. I really don't know. If you want to copy my life, don't do this. It's not a good idea. Also, I don't think that a model is good. I think that everybody should develop with its own character, wishes, environment, genetics, before it was modified and so on. I really don't know. I know that the key in science forget about the word success. The key in being a happy scientist is to do the work you love to do and try to answer a question that really, you really want to answer it. To go after the challenge of discovery. This is what, in my opinion, is success. If the group is larger and if the salary is larger and if you become a president, it's important, but there are other ways. And in my history, if you really look at it, was not smooth. Not at all.
Yeah.
So you show the protein in the ribosome actually change. I mean, in terms of copy number. So did you look into the RNA, the ribosomal RNA? Are they change as well?
Yeah, but there are actually many people look at that. And there are a few changes. Here a nucleotide, there a nucleotide. It's not something so striking. But maybe some of these changes are important. We cannot look at all now. We just started it. It means it's a starting project to look at ribosome under stress. We actually, I didn't say it, maybe I say it now. It's not only that we look at ribosomes in infections or in cancer. That's not correct. First of all, the first study was on healthy corn that didn't want to grow. It's not our work, but this was one of the motivations. But we look at every stress, heat, cold, magnetic field, whatever. And what I showed today, what I chose to show is two extreme situations under stress of disease, either influenza or cancer.
Okay. Professor Yang?
Yeah, thank you, Professor Jonas, for a fascinating talk. And I understand the last few slides, you actually put it up for us. Thank you very much. And I just have a very quick question and hope to talk to you the day after tomorrow again about the detailed possibility of collaboration. Now, when you see the variation in the ribosome copies for the cancer cell, and they may be somehow related to the heterogeneity, and what you're looking at are population of cells rather than individual cells. So is it possible that because of right now we know that there's a heterogeneity among individual cells within the population. So I think there's a possible that, I mean, when you want to enhance the efficiency of producing protein in those fast-growing tumor cells, you either build up more factory or you increase the production capability.
One of two.
Or maybe both, yeah. So
yeah, no, correct.
So
more of them faster or both. Yeah.
Yeah. So I think that would be certainly there may be some possible association with the individual cell heterogeneity.
About individual cell heterogeneity, it's very important, but I don't know exactly how I will do a copy number on that from the point of view of scientific possibilities, measuring possibilities. But what we are doing to somehow overcome it, we are taking a average. So we repeat the studies on either several tissues or several patients, and we look only for the changes that we see in all, which is of course not as accurate as what you want, but it's more doable. Maybe we go cell by cell with time, or with you.
Professor Ben-Susan?
You thank the Weizmann Institute in your talk, right?
Yeah, I should have thanked you too. Yes, you know, he was sitting in a committee that gave me a grant.
Yeah, okay. It was Michael Sella, in fact, the president. But you thank the Weizmann Institute, and in view of the introduction of the provost saying that CTU wants to go to life sciences and so on, what is the secret of the Weizmann Institute? How did you achieve, how this institute has achieved such fantastic achievement? And
well, I have to
of course,
to
you are one
you.
of them, but It's a fantastic place.
The present president is a physicist. And he said he doesn't care what a person is doing as long as he is very good scientist. Because a very good scientist will find the right problem to work on, even if when he comes, he has another idea. So this is one of the main points of the Weizmann, looking for best quality people. And then in my time, you want to know how I started. I was a chemist and I wanted to understand biological materials, and I thought that structure is very important, so I joined the Weizmann. Was the only place that there was some structure, of course not on proteins, but on fiber proteins like collagen, muscle. And the head of the department was an ex-student of Dorothy Hodgkin, which I mentioned earlier, in Oxford. Actually, he was a student together with another one, a woman that was kicked out because she was not good enough. You know her name? Margaret Thatcher. She was not good enough, but he was. And he finished, and he worked on vitamin B12, and he came to the Weizmann, and he started crystallography of small molecules in Weizmann. And he said, if somebody wants to do proteins, I will support. So I was the one. And he really supported me. When I came back from my postdoc, I was the only one that got an office for myself. All the other young scientists had to share offices. I didn't get money, but I got a possibility to ask for grants, for money, even not equipment. And I want to tell you, well, what was my office? This was a toilet that was at the end of the lab. Nobody used it because it was so far. He put a piece of wood on the seat and a piece of wood on the sink, and he said, this is your office. and I told him, it's wonderful because now I have a multi-usage office. And he said, no, the water is disconnected. So even from the toilet, one can get to do what one thinks is important in one's eyes. And this toilet is only a symbol, but most of my early life were not highly supported by the Weizmann, but full freedom I had. If I wanted to work on this, if I wanted to work somewhere else, if I wanted collaboration, everything that, once they trusted me, everything that I thought that is good for the project. Of course, this was the assumption. So I think this is one of the most important ingredients in Weizmann.
Okay. Thank you very much for your lecture. I'm a vet and I've worked in the field treating a lot of animals, and I've often been told that very few new classes of antibiotics have been produced in the last 20, 30 years. But actually looking at what you've just lectured, it looks as though many new classes of antibiotics could be discovered. Would that be true?
I fully agree with you. Very few were made. Actually, the last 15 years is the last. Sinarcid is the last one that came into use, I think 2002. Then linezolid 5 years earlier, and that's it. So I really agree with you that there is, I showed only very few examples. We have many more. And I think that the companies should go into it more seriously.
So let's thank again Professor Yonats.
Then to see what is there.
So, okay, thank you.